Dr. Bennett, you bred a virus to carry the gene, and Dr. Maguire, you injected it into a patient's eye, and there was light. So this is an incredible journey. I would like to start with a very, very basic question, Dr. Bennett. What is a person who has Leber's congenital amaurosis actually be able to see? And what causes this condition?
Sure. I'm going to start with a general answer, and then Al Maguire can be more specific. So there are at least 18 different genetic flavors of Leber's congenital amaurosis. Those are caused by different mutations in different genes. And depending upon the exact genetic cause, people have different levels of light perception versus vision early in life. And most of these genes actually cause damage or cause malfunction of the photoreceptors, and that explains the visual findings. So for the form that was just where we've been developing the gene therapy that just received FDA approval, that's a form caused by mutations in a gene called RPE65, and I'll pass it over to Al to describe what people can see with this condition.
Well, the RPE65 helps load nerve cells with the molecule that captures light called 11-cis retinol, and that's not functioning correctly in people with this particular form of Leber's amaurosis. They have some vision, and in some cases it's enough to get them, they're able to function and get around a bit when it's very, very bright. The way I think of it is they have about 1/10,000th to 1/50,000th the sensitivity to light that somebody with a normal retina would have. So it's as if you had a rheostat and you kept turning it down so that they are only able to use about 1/10,000th the light that you or I, and you can imagine that would make things difficult, especially when at hours of the day when it's not as bright out. It's sort of like a enforced curfew. There's also an issue with the central vision where there's some blurring because the resolution, the sharpness of vision in the center is reduced as well. So overall, it's dimmer and blurrier for people who are affected.
So Luxturna is approved for people who carry the RPE65 mutation. And essentially, we have a gene therapy which replaces this. So can you talk a little bit about what we will be exactly doing in the gene therapy here?
So, as you said, people who have this particular form of LCA would be candidates, and what is done, which is very similar to the protocol followed in the phase 3 trial, the efficacy trial, is we establish whether people have enough functioning cells that they're going to get some useful vision as a result of the treatment. If you have no cells, because over time they've wasted away, it really doesn't make sense to do the treatment. It's not for people who don't have functioning retina. So once it's established they have potential and enough cells, they undergo a procedure called vitrectomy, where the back of the eye behind the pupil is entered with tiny instruments, and The Luxturna, which is the gene therapy drug, is injected under the retina in the vicinity of the nerve cells, and those nerve cells will take up the drug. So that's done in one eye, and after anywhere from after 1 week, the other eye is treated as well. And while all this is being done with the surgery, the patient has a course of corticosteroids, something analogous to the dose you'd use for asthma or poison ivy, and that's to tamp down any possible immune response to the drug, since it is a modified virus. Fortunately, we really never have seen much, if any, response to the drug in terms of inflammation.
Mm-hmm.
And so just to sum up, yeah, sure. Go ahead.
Yes, Dr. Maguire just mentioned the virus. So Luxturna is also the first AAV viral vector product approved for clinical use. So Dr. Bennett, it would be really good if you could talk a little bit and explain what an AAV viral vector is for our audience.
Sure, and as you said, this is the first first viral vector that's approved for direct delivery for a genetic disease in the United States. And we're very excited about this. And so what this is essentially a shell. The virus typically has a shell of proteins which package the genetic material, in this case DNA. And viruses do what they do very well. They bind to cells and get into the cells, and then the DNA, or for other viruses, RNA, makes its way to the nucleus, where it takes up shop and starts encoding for the proteins that are encoded by that particular DNA strand. And so this recombinant virus is designed such that the bulk of the viral DNA that for the wild type virus, which can be copied in the cell, is removed, and instead it's replaced with the gene of interest, in this case of RNA, in this case RPE65, connected to a switch which turns it on, because without that the gene won't be expressed, and it's packaged in this shell, and so the virus is delivered to what's called the subretinal space. It's a space adjacent to the sick cells in the retina. It binds those cells, and then the DNA takes up shop in the nucleus. It starts producing the normal copy of the RP65 protein, and that corrects the biochemical deficit in those cells, which then allows them to function and allows the person to see.
That's incredible. We are talking about the viral vector, so I would also like to right now read out a couple of questions that have just come in regarding the use of the virus for the therapy. What happens to the copies of the bad genes that are being replaced in your eyes or bodies? That is one question that has just come in, Dr. Bennett.
Okay, so the bad genes stay there. In this particular disease, the bad genes are bad because they don't do anything. And so we don't need to remove them. They stay there, and instead these cells gain the new functioning genes, which take care of the missing function.
Okay. There's another one. Does the virus used in the gene therapy treatment make you sick in any other way? Does it?
So I'll handle that
does
one.
Does it do anything else to the body? Yeah.
Yeah. So that the one of the most important things that has been seen in AAV research, and that's not just eyes. That's systemically large organs in the body, where they're using amounts of this virus that are thousands of times more than what we're using in the eye. It's very, very safe. AAV, which is adeno-associated virus, is a recombinant or a genetically engineered parvovirus, which in humans does not cause any significant disease. There's some very esoteric condition called fifth disease, which most doctors have never seen, and it's really just a very mild viral illness. But that's the unmodified wild type adenovirus or adeno-associated virus. This has been basically any pathogenic or toxic factors have been spliced out of the virus, and that the virus is basically engineered so really all it does is it carries this piece of cargo, this new gene, into cells and does not produce any sort of toxin. So it is extraordinarily safe.
Thank you, Dr. Maguire. You already touched upon a little bit about the actual trial protocol. We're just trying to understand this a bit more. How much preparation should a patient or is a patient conducted through just before this injection or being treated with Luxturna?
Well, they're really, I mean, aside of the testing that's done before to make sure that they have the condition and they have enough viable cells to treat, they're really no different than any other person who's having a retina procedure in terms of the standard things: don't eat or drink after midnight or a few hours before the surgery, a general medical evaluation to make sure that there are no issues that might come up with the anesthesia, and really there's no significant difference, no major difference between any other type of eye procedure, eye surgery, and the Luxturna.
So how long does one procedure take? Like, for example, treating with Luxturna, what is the time for the surgery or the injection, that is?
So we know this well because we track times. So one way of putting it is in our original trial, it took about 2/3 the time it took to obtain an informed consent. The procedure takes about 50 minutes to perform from beginning to end. Certainly, the entire day, as I tell people, well, you have to show up to the hospital. We have to go through the checklist, and you have to stay afterwards for a few hours to make sure that you're properly positioned so that the drug can soak into the retina. But the actual surgery itself, about 50 minutes long.
And being an observer of all of these surgeries, I'd just like to add that whole time of surgery is mainly for preparing to do the injection, and the injection itself takes all of about 10 seconds.